So hi again, everybody. And great to see you all here. So I am going to spend a little bit of time talking about how everything that you just heard it can relate to Kabuki syndrome specifically, and what we're doing to move this forward. If I can advance my slides, I will. Okay, so, and first I want to say that all of this that we're about to talk about is the work of Dr. Bjornson and his lab, and this entire collaboration and clinical trial that we're about to hear is really thanks to him and his work. So I just wanted to acknowledge that up front. So Kabuki syndrome, as you heard Carlos talking a little bit, so KMT2D, just to use as an example, is a writer that places an open chromatin mark on the chromatin. So it allows the chromatin to open, makes the DNA available for making protein, and then making protein is at the end of the day what DNA does, because the proteins go and carry out the function in the cells. When you lose KMT2D, you get too much closed chromatin. KDM6A actually removes a closed chromatin mark. So again, the end effect of loss of KDM6A is too much closed chromatin. And so this is a normal balance. The balance in Kabuki syndrome is tipped towards way too much closed chromatin. And so the thought is that perhaps we can restore that balance with an epigenetic modifier. So you've heard throughout the 2 days of the conference from Dr. Bjornson, from Dr. Haldar, a little bit from Drs. Bodemer and Bonka about restoring this balance, and how pre-clinical trials have shown that this can be done. And so in addition, we know that the mouse models of Kabuki syndrome have, as again, you heard Dr. Bjornson talking about, and Dr. Haldar and others, these hippocampal memory deficits, and that we can test those in certain ways in the mice. And we see that the Kabuki mice perform more poorly all the time. We also know that the protein KMT2D, which binds with KDM6A, they're almost always found together, as you heard Dr. Kodani talk about, and Dr. Zippo, that it's highly expressed in the granule cell layer of the dentate gyrus, where this adult neurogenesis occurs. And then we know that deficits in adult neurogenesis lead to these hippocampal memory defects that we see. We know that they end up with a thinner granule cell layer, and we know that they have a deficiency of this neurogenesis in the granule cell layer of the dentate gyrus. Again, this is all things that you've heard a few times now before in these last 2 days of the conference. Now, Vafidemstat that you've just heard about is an LSD1 inhibitor. So it inhibits a protein of LSD1. So LSD1 removes an open chromatin mark. So it's an eraser, and it erases a mark that tells the chromatin to open. So even though it does not directly correct the absence of KMT2D, the thought is, if you lose KMT2D and you get too much closed chromatin, then maybe you can block something else that also leads to closed chromatin to restore that balance. And LSD1 is potentially that thing. And so in Dr. Bjornson's lab, this was published earlier this year in Molecular Therapy, you see that LSD1 inhibition does in fact change the histone methylation levels in the mouse hippocampus. This was not done with Vafidemstat directly, but was done with another LSD1 inhibitor that has the same mechanism. So, okay. And then this is just another way of showing this, that when you compare LSD1 inhibition in the Kabuki syndrome mice, what you see is that histone methylation that we saw disrupted is changed back to the way we see it in wild type mice. This is very exciting, but not enough, right? So the next question is, what about on the cellular level? What about that deficit in adult neurogenesis that we see in the Kabuki syndrome mice? And so the answer is yes. As you see on the bottom, so the top is with the plus means typical mice, the plus beta geo mean Kabuki mice. And what you see is lots of adult neurogenesis in the wild type mice, not so much in the Kabuki mice. And when given the LSD1 inhibitor, they look the same. It's restored. And here it is in a different way to look at it. So then the last step to this, for the animal models at least, is to say, okay, well, what about the actual defects we see in the mice as far as their ability to navigate visuospatial tasks as much as mice can do? And the answer is, again, we know that the Kabuki mice perform worse on this. And when they are given the drug, that in fact, again, they are restored back to the wild type mice. So that is significantly improved. So this data is extraordinarily promising that perhaps this can be the case in humans too, that LSD1 is correcting some of the molecular issues that we see in Kabuki syndrome, and that an LSD1 inhibitor can correct these. And so, of course, mice are not humans. And so the next step is to see whether or not this is the case in humans as well. And so this is why we're going to be launching HOPE, which is a randomized phase 1 and phase 2. Phase 1 is just kind of testing dosage, and we're doing that because we're going to include some participants that are under 18. And so that's not been done before with Vafidemstat, and so we need to do that testing. But then phase 2 is an early study for looking at efficacy, so whether or not this works. And so we are really, really excited about this. This is, could potentially be enormous for the Kabuki syndrome community. Again, the data that we have so far indicates that this is a really, really good route to pursue. And so we're very excited. And so I'm going to close with just saying that, I'm really, really grateful to everyone who has brought this project to where it is now and continues to work on it to bring it to you all. So again, Dr. Bjornson is doing a lot of work for this project, and he's been phenomenal, and it is his work that laid the groundwork for this. The Horizon team, I've worked with other industry partners before, and I can just say that they've been really, really fabulous. And they are extraordinarily receptive to feedback from the scientific community, from you all, the patient community, and because we can't do it without you, and that's been amazing, and I've really appreciated that. And then, of course, my amazing scientific collaborators outside of Hopkins and Kennedy Krieger, including Dr. Bodemer, Dr. Simpson, and others. And so we're really, really excited to announce this, and we will be able to bring you more details as soon as we have them, but we can potentially take some general questions.